Welcome to our cell project, and one of the things we just want to go over the cell project so you can listen to this over and over again at home. You can share it with your parents or anybody who's trying to help you with this project to better understand things. So we watched in class a video called Scale Ella from Math Snacks, and you can see that it has some different things going on with it. And so if you want to review that, it's just Math Snacks Scale Ella so that you can learn about scale factor. And what's happening here is we are going to be doing a cell, drawing it to scale and coloring it and labeling it and reflecting on it. And so a scale factor is what you're going to need to draw it by because it's a number that's multiplied to change the size. And we're going to be working in millimeters today. So I've blown up a little virtual ruler here. And you can see there are some inches and there are some millimeters. And so millimeters are on the same size as centimeters. And so you have 0 and 1 and 2 centimeters. And between each centimeter, you can count these little bitty marks here. And you'll see that there are 10 millimeters in a centimeter. And when you're drawing, you need to be very careful about how many millimeters everything is. Your teachers will be getting out a ruler and measuring, so make sure that if it's 1 millimeter, it's this big. If it's 2 millimeters, it's this big. Okay, and so on and so forth. So if it's 10 millimeters, then it should be this big, and so on and so forth. So make sure that you're very careful about how big you draw things because that's what we're looking for. So this is the paper that you're going to get in science class. And make sure you put your first and last name, what period you have math, what period you have science. We're both going to be grading it, and it's a test grade in both classes. You're going to be turning everything into Miss Kiki. So make sure that you have everything in by the deadline. And we're going to be enlarging the dimensions of a prokaryotic cell and a eukaryotic cell to scale. So you have to think about what numbers are going to work best for your scale factor. And I'll help you out with some formulas later on. So cells are so small, they're microscopic. So you're going to need to enlarge it by a really big number when you draw it so that you can see it. So you need to think about how you're going to keep those cells to scale. And so we have a checklist here to go through, and a lot of times you're going, oh my gosh, what do I do next? This is what you go to. So the first thing you're going to do is choose a scale factor for each cell. So whatever one you choose for the eukaryotic cell, use it for the whole eukaryotic cell. Whichever one you choose for the prokaryotic cell, use it for the whole prokaryotic cell. Okay? And so one of the questions is, do you want to use the same scale factor for both cells? We want to be able to see those organelles inside your cells. So make sure that you choose a scale factor that's going to do that for you. So you're going to enlarge each cell dimension using the math. And we have a whole table set up for you on that one. You're going to draw the prokaryotic cell using the ruler in millimeters to measure the sizes of the organelles and the cell parts. Make sure you're keeping it to scale. You're going to label those parts and then make sure you include a short description of what these things do, the flagellum, the pili, and the ribosomes. Above it, of course, you want to title it Prokaryotic Cell and color it neatly using colored pencils. Then you're going to move on to the eukaryotic cell. You're going to enlarge each cell dimension in that math table. Then you're going to draw it using your ruler in millimeters. And make sure you're keeping it to scale. Label the parts and include a short description of what these things do. The nucleus, mitochondria, endoplasmic reticulum, Golgi complex, and the ribosomes. And of course, your title should be eukaryotic cell. Color it neatly using colored pencils. And complete your math reflection by responding to these prompts. So you can handwrite it, or you can type it. It needs to be 10 to 15 sentences. So of course, sentences end with periods, and make sure you count those sentences. Then your science reflection is going to be in Google Classroom, so make sure you do that as well. So this is for the math reflection. So you're going to answer these questions in detail so that you have 10 to 15 statements. You're going to discuss why we needed to enlarge the original cell by a scale factor to be able to draw it. Discuss why you chose to multiply your original cell dimensions by the scale factor that you chose. So you're going to have one for prokaryotic cell and one for eukaryotic cell. So make sure you discuss how you chose those and why it was important. Then you're going to discuss the steps you took in showing your work for how the enlargement process worked by multiplying by a scale factor. So just here you're discussing the math. You're telling somebody who is absent and who doesn't know how to do this, or your little brother or sister, how you multiply. And you're going to make it connections to that base 10 number system because you're going to be multiplying by decimals. And how does that work with the base 10 decimal system? You're going to discuss the process of how you drew your cells and anything else. What did you learn? What can you take out of this activity? I'm not asking really for your feelings. I'm asking for the factual reflection. Next in your packet, you have the math chart. Notice first it's the prokaryotic cell. And the whole cell is 0.01 millimeters, super tiny. So you're going to have to multiply by a scale factor of your choosing. And you're going to do your work, probably at the bottom here, plenty of room. And you're going to write that new dimension. The new dimension is in millimeters, so make sure you put mm after each one of your answers. Do the same thing with the flagellum, the pili, the ribosome, and here, of course, it's just data unavailable. Then you're going to do the same thing with the eukaryotic cell. Whatever scale factor it is, it's the same scale factor for the whole eukaryotic cell. So you'll have the same number in all of these blanks. You're going to multiply the dimension by the scale factor. Show your work. A lot of students like to turn the page and work top to bottom this way. And then put your new dimension in millimeters. A little bit of extra room at the bottom if you need. Then we have some pictures. And this is a prokaryotic cell. And I have actually got a master's degree in biology. And this same picture is in all the textbooks. You can find it on Google, all that kind of good stuff. But you know what? We've discovered it's wrong. It's not to scale. So that's your job. Your job is to draw it to scale and to show us how big these things really are compared to each other appropriately. Same thing with the eukaryotic cell. So you can look at these and you can see, oh, well, that's the shape of a mitochondria. And this is how the endoplasmic reticulum works right here. But what we want to see is how big are they compared to everything else. And so you're going to be doing the math and the measurements to put all these items to scale. Now, when you're coloring these things, really the only thing that has to be a specific color is the chloroplast. And so hopefully you've learned in science enough about chloroplasts to know what color those have to be. Anything else can just be whatever color you'd like. So let's take a look at how we're going to do that. So we have 3 different types of paper that we have access to. We have some large construction paper, we have some poster boards, and we have some butcher paper. So I've gone ahead and measured those things across their smallest part so that you know what we're dealing with. Okay? So the general formula is you're going to take your cell size, multiply by your scale factor, and it needs to equal your paper size. So let's take a look at how this might work. For the prokaryotic cell, the whole cell is 0.01 millimeters. You need to come up with a scale factor after you choose what kind of paper you want to use. So you choose, if you want construction paper, 300 goes in here. If you want poster board, 500 goes in here. If you want to use butcher paper, 900 goes in here. So think about what kind of paper you want and then find the scale factor that allows you to draw the cell as large as possible. The eukaryotic cell is 1/2 millimeter. So once you figure out the kind of paper you're going to draw it on, you want to make it as big as possible. So put the paper number in here and figure out the biggest scale factor you can to fit that eukaryotic cell on that paper. So you're going to need to make sure and write these formulas, filling in our answers over here, and coming up with that scale factor. And it's going to be a big number because you see we're starting out with something super tiny. So Miss McKinney has tutoring, of course, every morning at 7:40 a.m. and I'm happy to help you with this. Most of it should be done at home, which is why I'm giving you this video to watch, and also anybody that's helping you with that can watch it as well. Have a great time, and I look forward to seeing your cells. Bye.